Lifespan: Why We Age and Why We Don't Have to, by David Sinclair. Live a longer, healthier life with new insights into aging. Aging affects everyone and can lead to discomfort, frustration, and eventually death. Historically, we have accepted aging as an unavoidable part of life. However, scientific advancements are changing our understanding of aging, with genetic research bringing us closer to identifying its root causes and potential treatments. Aging may seem inevitable, but it can be considered a treatable disease. Medical advances have transformed once deadly diseases into manageable conditions. What if we treated aging like a disease, like pneumonia or tuberculosis? Aging has various symptoms, such as dementia, organ failure, and loss of bone density, among others. As we age, our bodies take longer to heal, become more prone to infections, and experience reduced organ and brain function. Recent genetic breakthroughs suggest that aging is indeed a disease, one that could potentially be cured, adding years to our lives and improving our overall well-being. To treat aging effectively, we should target its root cause rather than focusing on individual symptoms. The history of cancer treatment provides a useful comparison. Until the 1960s, treatments were centered around removing cancerous cells without knowing or understanding the root cause, often damaging healthy cells in the process of this procedure. In the 1970s, researchers discovered the oncogene, the specific gene that, when mutated, causes cancer. This enabled targeted treatments that spared healthy cells much better and improved survival rates. Our approach to aging has been limited due to the lack of understanding about its root cause or causes. However, recent scientific breakthroughs are getting us closer to identifying it and potentially revolutionizing how we deal with aging. Our genes are designed for long life. The freshwater polyp Hydra vulgaris, a tiny water-dwelling organism, has long captivated geneticists because it has evolved to resist biological senescence. Senescence is the gradual decline of function that happens when cells stop dividing and accumulate in an organism, which is essentially the cause of aging. In the wild, Hydra vulgaris are exposed to predators and other environmental threats. However, under lab conditions, they appear to be virtually immortal, as they do not show any signs of aging. Other immortal creatures, such as the bristlecone tree, Greenland shark, and bowhead whale, have also seemingly evolved to resist or significantly delay biological senescence. Could humans evolve in a similar way? Our genetic makeup seems to suggest it is possible. While most living organisms have not evolved to resist senescence, they have all developed the same gene circuit, a closed system where one gene's action triggers another gene's reaction, and so on. To comprehend this gene circuit, we must go back to the beginning of biological life and the primordial soup filled with rapidly dividing cells. Scientists believe that these early cells contained two genes, which we can call gene A and gene B. Gene A is a caretaker gene that stops cell reproduction in response to environmental stress. After all, there's no point in reproducing in a hostile environment. Gene B is a silencing gene. When environmental conditions improve, it wraps gene A in a protein, turning it off and restarting cell reproduction. In simple terms, gene A is the red light for cell reproduction, while gene B is the green light. For some cells to survive, they evolve a gene circuit where gene B did not just turn off gene A, but also actively repaired any damaged DNA within the cell before reproduction resumed. This prevented the replication of damaged DNA, which is important because replicating damaged DNA can cause cells to lose their ability to reproduce. Cells that cannot reproduce become senescent. Today, humans display a more advanced version of this initial survival circuit. Its basic function remains the same, however: repairing DNA and preventing the loss of genetic information that leads to senescent cells and aging. In this sense, we are designed to avoid senescence. So, why aren't we immortal? The answer lies in that very same survival circuit. The information theory of aging suggests that aging results from the loss of cellular information. According to this theory, aging occurs due to a loss of cellular information caused by the very repair process meant to prevent aging. As we know, DNA can be damaged when repaired incorrectly, leading to a cell losing essential information for perfect replication, which ultimately causes the cell to stop functioning and reproducing. If our vital information were only contained in our DNA, this would not be an issue. DNA can replicate perfectly by itself. However, our epigenome is equally important. Our epigenome refers to all the processes and structures that regulate our genes and their expression. It can be thought of as the transmitter of our personal genetic information from the source, the fertilized human egg, to its receiver, our bodies. One of these epigenetic processes is gene marking, which contributes to information loss. Gene marking involves placing chemical tags on cells to identify their specific function, such as distinguishing a kidney cell from a brain cell. Some genes involved in this process, like sirtuin genes, play a crucial role in DNA repair, extending our lifespan and promoting vitality. These genes produce a family of proteins called sirtuins, which can move within a cell, turning other genes on and off in response to emergencies like illness or inflammation. Sirtuins act as roaming caretakers, making repairs and adjustments for each cell to remain healthy and functional. Longevity genes, like sirtuin genes, are incredibly important in combating aging. However, When they malfunction, they can contribute to aging. When sirtuins respond to emergencies, they may not return to their proper place within the cell. As unregulated agents, they can turn off genes that should be on, and vice versa. When sirtuins go rogue, they contribute to the loss of cellular information that causes aging. Boost your longevity genes to fight aging. While geneticists may be close to finding a cure for aging, people in some parts of the world have been defying the effects of aging for centuries. These areas, called blue zones, were first identified in the mid-2000s by author Dan Bettner, longevity expert Dr. Michael Pollan, and medical doctor Gianni Pes. Blue zones have unusually high rates of people in their 90s and 100s who remain active and healthy despite their age. So, what's their secret? Firstly, blue zone residents eat mostly plant-based diets low in animal protein. Consuming protein provides essential amino acids, the building blocks of all our internal proteins. These amino acids can only be obtained through diet, and animal proteins deliver them in large quantities. However, when we have enough amino acids, the body leaves survival mode, shutting down the genetic instinct to repair DNA damage and promote healthy cellular replication, which is triggered during stress. Plant-based proteins, on the other hand, supply just enough amino acids to maintain healthy bodily functions while still keeping the body in a state of beneficial stress. This level of stress keeps the survival instinct to repair and preserve DNA active without compromising overall health and function. Another thing blue zone residents do to maintain a healthy level of bodily stress is restrict their food intake. A 1978 study showed that residents of Okinawa Island, a blue zone, consumed 20% fewer calories than mainland Japanese people. In Aigara, Greece, residents follow a Greek Orthodox calendar, which includes extended periods of fasting. Fasting is believed to be another simple way to create the beneficial stress that activates longevity genes without overworking them. In 1935, researchers at Cornell University even demonstrated that rats with restricted diets lived significantly longer. This study has been confirmed over and over. Long-term caloric restriction might not be suitable for everyone. Fortunately, a 2011 study from Yeshiva University found that intermittent fasting, where participants fasted for 5 days a month, can achieve the same effects as long-term fasting. After 3 months of intermittent fasting, study participants reported lower levels of the hormone IGF-1, insulin growth factor 1. High levels of IGF-1 are thought to contribute to aging and disease, while geneticists Nir Barizali found that families with women who routinely live past 100 years old have lower levels of IGF-1 in common. An aging cure might be closer than we think. A world without aging might seem like a fantasy, But geneticists are working hard to turn that fantasy into a reality. Interestingly, many anti-aging treatments come from the natural world, including soil bacteria and wildflowers. These natural sources might hold the secrets to curing aging. One potential cure originated in Rapa Nui, an island off the coast of Chile, also known as Easter Island. In the 1960s, scientists discovered a unique bacteria in the soil beneath one of the island's famous head statues. This bacteria contained an antifungal compound called rapamycin, initially used to suppress the immune system to prevent organ transplant rejection. Rapamycin was later found to have life-extending potential. Studies showed that fruit flies given rapamycin lived up to 5% longer. Mice near the end of their natural lives who were fed rapamycin lived 9 to 14% longer than expected. Rapamycin works by inhibiting the longevity gene mTOR, or mammalian target of rapamycin. Although mTOR promotes cellular growth when functioning properly, it can go rogue as we age, directing cells to stop dividing. Rapamycin helps regulate mTOR genes so they don't work against us. While more research is needed on rapamycin's effect on humans, another anti-aging drug is already prescribed for diabetics: metformin, which comes from a wildflower called goat's rue. Metformin activates an enzyme called AMPK, which restores mitochondrial activity. Mitochondria are tiny cell structures that convert nutrients into energy. Stimulating them increases the energy available for cellular repair and function. Metformin also inhibits the metabolism of cancer cells. A 2017 study found that regular metformin doses in elderly humans reduced dementia by 4%, heart disease by 19%, and frailty, an age-related syndrome with symptoms like weakness, unintentional weight loss, and exhaustion, by 24%. In our lifetime, metformin, rapamycin, and other aging antidotes under development might be widely prescribed to prevent aging and extend vitality in old age. Meanwhile, researchers are also fighting aging by targeting senescent cells, also known as zombie cells. To combat aging, we need to tackle zombie cells. As time passes, our cells become senescent. Their DNA gets damaged, and they lose the information required for replication. Worse, the accumulation of senescent cells accelerates the aging process. Although these cells no longer function, they are not entirely dead. Instead of disappearing quietly, they send out distress signals, causing a false state of emergency. This makes neighboring cells also become senescent, further speeding up aging in the body. Zombie cells also release cytokines, proteins that cause inflammation, which is linked to age-related illnesses like heart disease and dementia. DNA damage can occur in several ways. Anatomist Leonard Hayflick suggests that one way is through telomere shortening. Telomeres are protective caps at the end of each cell's DNA sequence. Hayflick observed that as human cells divide, their telomeres become shorter, eventually exposing the DNA itself. When the DNA strand is exposed, two things can happen. The cell may interpret this as a break in the DNA chain and try to repair it by rejoining the broken chain. However, incorrect DNA refusion can cause the cell to become cancerous. Alternatively, the epigenome, a complex system of chemical compounds and proteins regulating our genes, may intervene. It can signal for sirtuins to shut down the cell completely. This prevents the formation of cancerous cells, but also leads to a buildup of these senescent zombie cells. How can we tackle this zombie cell protein? It's a complex issue. We must eliminate senescent cells without harming the rest of our delicate cellular ecosystem. Senolytics, a new class of pharmaceuticals, might be the answer. Senolytics are drugs designed to target and eliminate senescent cells specifically. Researchers at the Mayo Clinic have tested two senolytic molecules on mice: quercetin, found in kale, capers, and red onions, and dasatinib, a chemotherapy drug. Mice treated with senolytics had an average lifespan increase of 36%. While these results are far from conclusive, they are very promising, and the potential for senolytics to fight aging in humans is enormous. There might be a way to reset aging. In 1996, Dolly the sheep was cloned from a single adult sheep cell, and the world was amazed. However, for some scientists, the most fascinating aspect of Dolly was the cell from which she was cloned. Dolly was created using an older sheep's cell, which meant that old cells could generate young cells. Even old DNA still contained the blueprint for youth. So, can we reverse aging and hit the reset button? It is possible. As we know, aging happens through the loss of cellular information. This loss can be significant. Scientists estimate that we lose about 80% of our original cellular information throughout our lives. Cloning experiments have shown that not only is this original information stored within our DNA, but it can also potentially be unlocked. Japanese scientist Shinya Yamanaka may have found the key. Yamanaka identified 4 genes that can transform into pluripotent stem cells, or young cells that have not yet been assigned a function by the epigenome. These 4 genes, called Yamanaka factors, can potentially be reset to perform any function in the body. Researchers at Harvard have already observed promising results from reprogramming Yamanaka factor genes in mice. What would life look like if we manage to reprogram these genes in humans? We might receive an injection containing the Yamanaka factors in our 30s, with the virus lying dormant until our 40s. Then we'd get another injection containing a molecule like doxycycline, which would activate the Yamanaka factors and initiate cell reprogramming. We would undergo this process periodically in our 60s, 80s, and possibly beyond 100 years old. This raises another question. What would be the impact on our world if the average lifespan extended well over 100 years? A longer living population might be beneficial. In the book The 100-Year Life, it's stated that half of the children born in Japan in 2019 will live to be 107, while half of those born in the United States in 2019 will reach 104. As scientists get closer to finding a solution for aging, our longevity predictions continue to improve. For each month we live, we gain an extra week to our lifespans. But what will the world look like when people are routinely living beyond a century? We already have an aging population, and elderly individuals who are retired can be expensive. They no longer contribute to the economy through work, and the costs of their health care and social benefits continue to rise. Won't a higher percentage of people living longer put more pressure on an already strained system and world? It actually depends on how our lifespans are extended. If we can not only lengthen life, but also conquer the problem of aging, our future might be much brighter. Economists at the University of Southern California suggest that since delayed aging would allow people to continue working, that would actually contribute to the GDP, and in the United States alone could profit up to $7 trillion by 2069. This is supported by a 2009 European Union report showing that countries with lower retirement ages have lower GDPs. When people can work longer, their spending power also increases, also contributing to the economy. After retirement, individuals generally spend less and make fewer significant investments. With extended lifespans and increased vitality, potentially trillions of dollars in unspent retirement savings could be unlocked and injected into the economy. Curing aging will likely boost the global economy, but the benefits do not end there. As a society, we would all be in the position to lead more energetic, productive, healthy, and happy lives. Once we fully understand the positive impacts of eradicating the problem of aging, perhaps we will join leading geneticists in wholeheartedly seeking its cure. To enjoy longer, healthier, and improved lives, we must change our perspective on aging. Aging is not an inevitable part of life. Rather, it is a condition, a disease that can be studied and potentially cured. Genetic researchers are steadily making progress in reversing the aging process, and we may soon find ourselves in a world where aging is eliminated and people live much longer than before. This concludes Lifespan.